today's urine analysis practical, let us learn the chemical examination of urine with the help of chemical tests and learn to interpret the findings obtained in the urine analysis. Benedict's test is performed to detect reducing substances in urine, which are glucose, lactose, maltose, fructose and galactose. Now, Mr. Sujeeva will demonstrate how Benedict's test is performed. To 2.5 ml of Benedict's reagent, add 4 drops of urine. Boil for 2 minutes over a flame or place the tube in a boiling water bath for 5 minutes. Allow to cool slowly and observe for color change or precipitate formed. Now, let us learn how to interpret Benedict's test. It is a semi-quantitative test. So, if the specimen upon boiling changes the color to a green color, it signifies there is traces of reducing sugar amounting to 0.2 to 0.5 gram per deciliter. And if the color changes to yellow precipitate upon boiling, it signifies traces of reducing sugar present, which can amount to around 0.5 to 1 gram per deciliter of reducing sugar. If you get an orange red precipitate, it signifies there is moderate amount of reducing substances present, which can quantitate to around 1 to 2 gram per deciliter. And brick red precipitate signifies a large amount of reducing sugar present, which can amount to more than 2 gram per deciliter. Our specimen showed a yellow precipitate, signifying that there is trace amounts of sugar present. Rothera's test is performed to identify ketone bodies in urine. To 5 ml of urine, add crystals of ammonium sulfate and mix until saturation is achieved. Add 8 drops of 5% freshly prepared sodium nitroprusside solution and mix. Lay along the test tube wall 1 ml of concentrated ammonia solution without shaking. A purple ring at the junction of liquids indicates the presence of ketone bodies. You can appreciate that the sample is positive for ketone bodies. Rothera's test detects only acetone and acetoacetate. Moving on to heat precipitation test, which is performed to detect proteins in urine. Fill 3/4 of a test tube with the urine sample. Heat the top 1/3 of the urine in the test tube to boil. Observe the change. Any increase in the turbidity in the upper 1/3 compared to the lower unheated part indicates the presence of proteins and phosphates. When about 3 drops of 10% acetic acid is added, turbidity due to phosphate disappears. If turbidity remains, it indicates the presence of proteins. You can appreciate that the sample is positive for proteins. Another test to detect proteins in urine is the sulfosalicylic acid test. This test is performed by mixing 1 ml of centrifuged urine with 2 to 3 drops of 3% sulfosalicylic acid. Turbidity indicates presence of proteins. The degree of turbidity is an estimation of the quantity of proteins present in the urine sample. This chart shows the degree of turbidity. You can appreciate if there is trace amounts of proteins present, a rough value of proteins present can be around 6 to 30 milligram per deciliter. In this tube, there is distinct turbidity with no granulation, roughly quantitating to around 30 to 100 milligram per deciliter of protein. In this, there is turbidity with granulation with no flocculation, and this roughly quantitates to around 100 to 200 milligram per deciliter of protein. And this you can see turbidity with granulation and flocculation, which roughly quantitates to around 200 to 400 milligram per deciliter of protein. And here you can appreciate clumps of protein, which quantitates to a protein of more than 400 milligram per deciliter. Appearance of conjugate bilirubin in urine can provide an early indication of liver disease. Foucher's test is performed to detect conjugate bilirubin in urine. This test is performed by adding 1 ml of 10% barium chloride to 2 ml of acidified urine. It is mixed and filtered, and this is spread out the precipitate on the filter paper and dry over a Bunsen flame, or dry by placing it on a second dry filter paper. Place a drop of Foucher's reagent on the precipitate. Observe the color. Green color indicates the presence of bile pigments. Reagent strips currently provide a simple and rapid means for performing medically significant chemical analysis of urine for a number of analytes. These reagent strips consist of chemical impregnated absorbent pads attached to a plastic strip. Let us look at the technique of using this strip. First, dip the strip briefly into a well mixed uncentrifuged urine specimen at room temperature. Remove excess urine by touching the edge of the strip to the container as the strip is withdrawn. Blot the edge of the strip on a disposable absorbent pad. Wait the specified amount of time for the reaction to occur. This is given by the manufacturer. Compare the color reaction of the strip pads to the manufacturer's color chart in good lighting. You can see that in this reagent strip, the proteins are positive. There is 1 plus for proteins. Blood may be present in the urine either in the form of intact red blood cells as in hematuria, or as the product of red blood cell destruction, hemoglobinuria. Next, we will see how to distinguish hematuria from hemoglobinuria. In both hematuria and hemoglobinuria, before centrifugation, the urine is red in color. After centrifugation, if red cells settle at the bottom of the tube, leaving a clear yellow urine supernatant, it is due to hematuria. While if the urine sample remains red even after centrifugation, it is due to hemoglobinuria. Tea-colored urine can be due to the presence of certain substances present. These include bile, bile pigments, bilirubin, certain dyes and drugs.